This video lecture is going to finish out chapter 25 part 2. I'm going to start on slide 12 of these PowerPoint slides. So we're going to go through some different examples of DNA lesions that can occur and how they are repaired by different types of repair systems. One example of a DNA lesion is deamination, and this is the process in which there can be spontaneous hydrolysis of an amino group on the amine of a base, and when this happens, this leaves a carbonyl in its place. So for example, if we look on the top right part of this figure, we can see cytosine. It has an amino group on it. It can become deaminated, and when it does so, it becomes a uracil. Now you might wonder, why is it that DNA contains thymine and not uracil? Recall that DNA has thymine and RNA has uracil, and both uracil and thymines can base pair with adenine. The only difference between uracil and thymine is the fact that thymine has a methyl group attached to carbon 5 of the pyrimidine ring. One of the most common spontaneous DNA lesions is the natural deamination of cytosine to uracil. And so uracil is going to base pair to A instead of G. And as a result, it ends up where there should have been a GC base pair is now an AT base pair. Fortunately, uracil is an unnatural base in DNA, and DNA polymerases they are able to recognize that as a mistake and replace it with the correct base. Another example of a DNA lesion is oxidation of a base. Example is guanine can become oxidized to form 8-oxoguanine, and then 8-oxoguanine can form base pairs with adenine. Another lesion is depurinization. So depurinization is the process in which you can have hydrolysis of the base from a purine nucleotide in RNA and DNA, and this results in an apurinic residue. There's a specific type of repair mechanism called base excision repair that is able to repair a lesion by removing the base at a site. So imagine that you have a base that's damaged by oxidation or chemical modification. In base excision repair, an enzyme called DNA glycosylase will actually remove the purine or pyrimidine within the DNA and leave the backbone intact. So DNA glycosylase will leave what's called an abasic site because it's apurinic or apyrimidinic, where the base sugar bond is cleaved. And then a different enzyme called AP endonuclease will come in and cut out a short stretch of DNA around that AP site, leaving a gap. And so then DNA polymerase 1 will come in and fill in that gap, and the gap will then be sealed by DNA ligase. Another example of a DNA lesion is thymine dimers. Ultraviolet light can cause the pi bond of adjacent thymines to rearrange into sigma bond, forming a cyclobutane structure. So essentially, there is a covalent bond between two adjacent thymine residues in a strand of DNA. This thymine dimer causes a kink or distortion in the molecule. So nucleotide excision repair is one of the mechanisms that are used to repair these lesions that are caused by either ultraviolet light or chemical means, which leads to some type of deformity or distortion in the DNA structure. Exin nuclease recognizes that distorted DNA that's caused by the lesion. It will then cut 2 phosphodiester bonds, one on each side of the lesion, and then the excised nucleotides are then removed, and then DNA polymerase 1 will come in and fill in that gap, and then the nicks that are remaining will be sealed by DNA ligase. There are several types of damages that are repaired without removing a nucleotide or base, and this is called direct repair. For example, these thymine dimers can also be repaired by direct repair in addition to nucleotide excision repair. These enzymes called DNA photolyases, they are going to use the energy derived from absorbed light to then reverse pyrimidine dimers. Another direct repair example is the dealkylation of bases. So guanine can become methylated into O-methylguanine, and that methylation of guanine can cause incorrect base pairing. So it can cause that methylated guanine to base pair with thymine. There are enzymes that can specifically dealkylate the methylated guanine by transferring that methyl group onto the cysteine of the residue and reversing the alkylation to give back the guanine nucleotide. So let's talk about double-stranded breaks and how that is repaired in the cell. Many types of DNA damage result in a single-stranded break. Now, if there is a single-stranded break that happens during replication, then if a replication fork encounters a single-stranded break, it will be converted into a double-stranded break. And double-stranded breaks are actually pretty bad news, but they can be repaired by this mechanism called homologous recombination, as long as there's another copy of the DNA in the cell that is very similar to it, as there would be if the DNA had just been replicated. So homologous recombination is a mechanism of repair in which it repairs a double-stranded break, and it does so by combining two molecules that are very similar to each other, so they have homologous sequences. So during replication, a nick in the DNA can cause a double-stranded break, and this will cause the replication fork to collapse, and so homologous recombination is then going to be used to repair that double-stranded break. So the first thing that happens is the 5' end at that double-stranded break is degraded. This is going to then result in a 3' single-stranded extension, or a 3' overhang. And then an enzyme called DNA recombinase, it's actually a set of enzymes, is going to actually use that 3' single-stranded piece of DNA to then invade into another strand of DNA that's homologous to it to promote strand invasion. And so the 3' end is going to invade with an intact homologous DNA duplex and pair with it its complementary sequence. And this is then going to create a branch structure, which is the point where you have 3 DNA segments that come together. So once we have strand invasion and formation of the branch structure, then we have what's called branch migration. And so in this case, the branch will migrate to the left of the break, and then the other strand of DNA that was originally broken is going to pair with the homologous DNA strand, which is then going to create this Holiday intermediate, which is this crossover structure. And then the Holiday restructure will then become resolved by cleaving it by using a special type of nuclease, followed by ligation of the nicked DNA. And so the overall process is going to then reconstruct the replication fork. And so this is an electron micrograph of a Holiday junction. of DNA that is in the middle of undergoing recombination repair. Homologous recombination is important for DNA repair, and it's also a very important mechanism for allowing recombination to occur during mitosis. We've just talked about how homologous recombination can repair double-stranded breaks. There's also another mechanism of repairing double-stranded breaks called non-homologous end joining, otherwise known as NHEJ. Homologous recombination requires another strand of DNA that is similar to the broken strand. Non-homologous end joining is more flexible because it does not require a similar second strand of DNA. And in non-homologous end joining, it involves Cu proteins, which then bring two pieces of DNA together, and then DNA ligase will come in and seal the DNA, so it'll form phosphoester bonds between the two DNA strands. However, the problem with NHEJ is that it's potentially less accurate because you could actually have the wrong ends that could be joined together, or in some cases processing of the ends can sometimes result in deletion.